Okay, hi everyone. It's 3 o'clock, so I'm going to go ahead and get started. It is my pleasure and honor to introduce Dr. Tina Lasisi. Tina is coming to us from the Department of Anthropology at the University of Michigan. She got her start at the University of Cambridge in England, where she majored and got a BA in archaeology and anthropology, and got first class honors with a U. I'm not sure what that means. She then did her PhD at Penn State with Nina Jablonski and Mark Schreiber in biological anthropology. She did a very brief postdoc at Penn State, also with Nina, who maybe only advises people who have rhyming names.
Yep, exactly. We're a team.
Yep. And then did another postdoc at USC, University of Southern California, with Doc Edge and Jaslyn Mooney. And she is now an assistant professor here. I'm super excited about her work. You may also know her, in addition from being on campus, from her scientific communication outreach. She is the host and a writer on a PBS show called Why Am I Like This?
Got it.
And she's also very active on like the TikToks and Instagram, and does a lot of work in that arena as well. And you may also know her from her recent PNAS paper, in which she used a thermal mannequin to determine what the biological properties or thermal properties of different types of human hair are. I don't know if she'll be talking about that in this, but if you're interested, you should definitely ask her about it. So with that, I'm going to turn it over to Tina. Please give her a warm EEB welcome.
Okay, thanks everybody. It's so exciting to be here. Ever since Midwest Pop Gen, I was like, oh, I wanna be friends. I wanna sit at the cool table, and I feel like I've made it now. So I'm gonna be talking to you about the work that I do on hair and skin, and I did not include the thermoregulation stuff that I did, because otherwise you would be here for like an hour and a half. But please do ask me questions. I have pictures of Terminator-looking robots called thermal mannequins. But with that, I think that there are a few phrases that confuse people as much as race is a social construct, and yet it's a phrase that's often offered as an explanation when people ask about human differences. Biological anthropologists often find themselves involved in conversations about what race is and is not. And depending on who you ask, we are either partially or entirely responsible for the concept of race, so I don't know. Now, the discipline of anthropology is one of the ones that is doing the most to backpedal from what our predecessors did, which I'm sure you will know from your disciplines as well. Sometimes people in your academic lineage do messed up things, and now you have to clean it up. So, since the 20th century, anthropologists, among many others, have been making statements on the non-existence of race. And not too long ago, I was actually involved in making one of these statements. But to be completely honest with you, I am now really questioning what the point is of making statements like these. Because people still talk about race. Race still persists, even in science. And the question is why? Over the years, I've heard a lot of people talk about the reasons why race still persists. And people will say that it's a question of education, that we haven't quite figured out the right way to explain to people that race isn't real. But I don't think that that's true. And like I said, the reason I don't think it's true is because race persists within science as well. Whether it's explicit or wrapped up in the euphemistic ancestry, ethnicity, population, we see these groupings that echo racial categories everywhere from clinical studies to forensics. So to put it simply, I don't think that the issue is a breakdown in communication. I don't think that we are failing to transmit the correct information so that race is no longer an issue. I think that the issue is much deeper, and it's about how we're doing the science. Specifically, I think we have a phenotyping problem. If we don't critically examine the way that we use phenotypic categories, we are doomed to recreate race over and over again, no matter how hard we try to rebrand. Two issues that I see illustrated in hair and skin are the distortion of variation within a phenotype and the creation of composite traits, so kind of grouping and lumping things together that should better be studied as distinct traits. Part of the solution is the use of objective and biologically informed phenotyping methods. Skin color. It's the quintessential story of what can be gained from using good phenotyping methods, objective ones, and actually measuring something. Reflectance spectrophotometers are the gold standard for measuring skin pigmentation. These are instruments that are able to give us, among other measures, something that we just describe as melanin index, or M-index, which is a proxy for the melanin content in someone's skin. But as great as this is, and as widely as this is used, this wasn't always the way we talked about skin color. An earlier way of discussing skin color variation involved the literal assignment of colors to races. I will at some point circle back about how Linnaeus is to blame for everything, but for now, suffice to say that this simplification of human groups into different types is limiting for a lot of reasons, but especially because it glosses over the variation within groups. And yes, you can learn a lot from comparing and contrasting groups. In fact, it's how we've learned about some of the fixed alleles that explain skin color differences in populations that have been separated for a very long time. But you also miss a lot of variation if you are only thinking about comparing and contrasting groups. If it wasn't for the use of melanin index, we wouldn't have been able to understand the genetic architecture of pigmentation within the African continent. In fact, for the longest time in the molecular literature on skin color, a lot of the discourse focused on the lack of variation in African populations in the MC1R locus. In contrast, Europeans were highly polymorphic at MC1R. And even though these papers are like 20 years old, I think it's really worthwhile to unpack why this happened, because it reflects very long-standing historical views on pigmentation and the nature of variability among human populations. And something that I find pretty striking is the recurrent discussion of pigmentation as this body-wide thing where you're talking about skin color and hair, right? Like, we're talking about pigmentation, but if you look here, people are talking about red hair and light brown hair, and you start to wonder, okay, well, you're telling me about pigmentation writ large, but you're only really looking at different visible hair colors, which obviously is easier to do in European populations. In fact, the variants that we think about when we're talking about MC1R variation, a lot of them have effects on hair specifically. That's what we know the largest effects to be. So diving further into the methods of this paper, we see that they used a wide range of European populations, and not a super wide range of African populations. And they also mentioned African Americans. So that makes us wonder a little bit about, like, what are the groups that you're defining? Where are you looking for that variation? And something that was strange for me to see is how they define Asia, because I had never seen Japan, Papua New Guinea, and southern India put together as a category, but that just goes to show, like, how subjective, and even within our super scientific ways of thinking about human genetic and phenotypic variation, we've made a lot of subjective groupings that affect our analyses and the results that we get. So the theme of the literature here is the variable nature of Europeans and the homogeneity of others. It's not really controversial to say that genetic research has historically overrepresented Europeans to the detriment of other populations. But what I think is often missed in these discussions is that phenotyping really matters. The methods that we use strongly influence our ability to detect certain kinds of genetic variation. That's why these 2017 papers on African skin pigmentation from Sarah Tishkoff and Brenna Henn's labs made such a huge impact. They both countered this widespread narrative that Africans were homogeneous in pigmentation and underlying genetic variation relating to pigmentation. And of course, there's a little bit of variability between what the broader public thought and what people who are really in these disciplines understood. But the number of headlines back then that were along the lines of, surprise, Africans are diverse in skin color. We didn't know this. How could we have known? And what I think is interesting is that it's not just a fun debunking of a Eurocentric view, but it's a scientific victory in being able to discern variation that was previously invisible. And that's a leap forward in our ability to understand the evolutionary history of a complex trait. Think about how much more complex the story becomes when you stop saying, okay, humans left Africa and then they changed skin color, and you start thinking about all of the history that actually was occurring in the African continent and didn't just stop after certain groups left the continent. So that's skin color. But how did we talk about hair, and how has that changed? This is a map here by the Italian geographer Renato Biasuti. I always go back to these maps. Like, sometimes people in the past, the problematic past, be it as it may, had really good graphics, and sometimes you got to go back to them. And this is like a very, physical anthropology thing. Like, let's have a map, and like, what's the distribution of variation in the world? There's a lot of these things from, the 19th century up until like the mid-20th century where people are trying to just say, okay, here's the distribution of variation. And this is all based on broadly defined ethnographic data. So what I have gathered that means is that he kind of went through travelers' descriptions of what different populations looked like and was like, okay, this is what the populations there looked like. Now, in the legend, we see terms that are still often used to describe hair. Some of these are a continuum of curvature, from straight to wavy. Others are describing cross-sectional properties, like flat, cylindrical, or oval. And others yet seem randomly sprinkled in. Like you see short, which I can only assume is the length of the hair, and coarse, which is something that is really associated with like how thick the hair is. But something that's really interesting to note here, and we're going to go back to, is how these traits are paired together, right? These composites, like curly hair is always going to be short and flat in cross-section. Now you might think that this is from the past and we've gotten over it. It's not. Hair is still talked about in this way in a lot of different disciplines, like forensics, where hair is often used as trace evidence. One of the analyses that was carried out, is it still being carried out? That's a whole other story, is this determination of racial origin. So you have this mystery trace evidence, like first step, narrow down the suspect list. What's the racial origin? Good scientific question. The way they do this, and I'm happy to go into deep, deep detail, qualitative assessments are made by an expert who is an expert in judging these characteristics and can say, oh, this is definitely like this racial origin or that racial origin based off of my expert experience. Now, another field that often uses racialized approaches to hair is dermatology. And we can talk about medicine, but let's just, dermatology specifically, skin, hair, that's their lane. And you can see here that the different descriptions align very closely with what you saw in that map. You see that people whose quote unquote biological race is black have frizzy hair that has flat cross sections. Oriental people are so invariable in having straight hair that it suffices to have one sample. And white people are uniquely diverse in having a range of hair textures and hair colors. I'm joking, but there is really an absence of vocabulary to describe the range of variation beyond Europeans. And so that is something that you run into with a lot of these classification systems where it can become invisible, where the limitations of language can become invisible. So if you consider hair phenotyping in genetic context, you might think, okay, well, geneticists are smart. They know to do better than this. A lot of them categorize hair as straight, wavy, or curly, which is what we do in everyday life as well. A recent meta-analysis on hair GWAS did this in about 5 of the 8 studies that they overviewed. Also, I'm so sorry, I just said recent and then I saw 2018 and I just had a moment. It's fine, I'm good. There's another one of the papers that was included there that included a 4th category of frizzy. There was one that divided wavy into slightly wavy and wavy, and then there was another that had big curls and small curls. It doesn't matter how many nuances and adjectives you add to this, it's still a subjective judgment of like, where does small curls start, where does frizzy begin? Nobody knows but the person who is deciding. So if we want to quantitatively phenotype hair, we got to start in thinking at what level of morphology we're interested in. So you have the entire scalp. That's usually where these subjective qualitative descriptions lie. It's, okay, is it wavy, curly, based on the whole scalp? You can think about the individual hair fiber. You can even look at the microstructure of hair, and you can even analyze it on a molecular level. My work has mainly focused on the individual hair fiber, looking at cross-section and curvature. We're about to go really deep into methods. I mean, I don't know if you're excited. I know there's one person here who asked me about my hair phenotyping methods, so this is for you. Shout out if you're here. Hair curvature describes the spectrum from straight to tightly curled hair. So it's a way of operationalizing it on the level of a single hair. How much does it deviate from straightness? Then there's the cross-sectional morphology that includes both variation in eccentricity, so the range from round to flat, but you can also think about the cross-sectional area. So that's kind of where people are talking about fine and coarse hair. Really, they're just talking about how thick the hair fiber is, which sounds less, I don't know, fancy, I guess. So, surely I'm not the first person to think about this. Like, trust me, my entire career has been like, I feel like I'm saying something so obvious here that someone is gonna pull up and be like, look, these people have been doing it and you just don't know how to Google things. But the question has always been for me, if classifying is so clearly bad, why aren't more people measuring? It seemed so simple to me. I remember it like it was yesterday. I started as an undergrad and I was like, I'm gonna solve this. Well, it turns out that it's really difficult. That's why. So let's start with cross sections. To see the cross section of a hair, you first need to slice the shaft. Use a microscope to look at its cross section. Now, this isn't fundamentally difficult. We've been doing it since the 19th century, actually, which is why you can find, fun old papers describing observed cross-sectional differences between races of man. Hair is a race characters, bars, that's funny. So I did say that most GWAS were doing a bad job, basically. But there's one study that I really loved, was a big fan of, from 2008 in Human Molecular Genetics, where the authors describe an association between the ectodisplacent A receptor gene, EDAR for short, and hair thickness in Asian populations. I thought it was such a great paper because I was like, look, this person did a thing. They actually measured stuff and they found things. Like, they're such a great example. So many eons ago, when I was just starting out my PhD, I figured, why don't I just email the lead author? Maybe they have tips for dealing with cross sections, because I actually couldn't figure out how to replicate things from their methods. This has been a theme in my life. Method section, not enough detail. But I emailed him, went ahead, forgot about it, and 9 months later, he replied. The lesson here is it's never too late to reply. I know that there are emails you still need to get back to, and if you're waiting for one, don't give up hope. It might happen. So, Akihiro was very kind to dig up this stuff from his archives, as it was literally a decade ago that he had done this work, but he found a slide of what their methods were. So on the top row, you see this embedding process for the hairs in paraffin, where the hair is basically, this is the problem with paraffin. Paraffin is like a wax. I don't know how many of you embed things. So it's soft. And hair is just the worst combination of characteristics. It's super long, but it's really got a very small cross-section, so it's difficult to handle. And it's also really hard. And so you put in something like paraffin, and you slice the cross-section, you can see that it moves. So paraffin is not really great if you want to make sure you're getting good cross-sections. And on the bottom, they basically used epoxy. And you get a nice, clean cross-section. So that's nice. But the issue with using epoxy instead of paraffin, either of them, is they're both finicky. With epoxy specifically, it takes up to 24 hours or longer to cure or dry. And then the second part of this is curly hairs. That's what I'm interested in. They are super hard to embed because you basically need to make sure that your curly hair is stretched out straight while it is curing in epoxy over 20, how? I tried a lot of different ways and none of them were great. And so things would end up getting messed up. It was just like a lot of effort. And Akihiro agreed. He said, yeah, it's really hard. Luckily, curly hair is pretty rare in Japan and Southeast Asia. So, pick your populations better. Make sure they have phenotypes that are easy to measure. So, after a lot of trial and error, different techniques I tried, I developed a method using a low melt point plastic called polycaprolactone, where you can heat it up, honestly, basically at body temperature, and then you can instantly embed the hair. So you can stretch it out and it's immediately like captured and in the plastic, and then it can dry immediately. And so you have this nice straight hair that you can get a clean cross section on. And then on top of that, super great benefit is that the final product is soft enough to just cut with a razor. So that's one of the benefits of paraffin. It's like easier to slice. With epoxy, you sometimes need like diamond tipped something, and when you hear diamond tipped, you know your budget is already crying. So I am presenting this in like 2 minutes. This was 6 years of my life. Just a moment of silence for that. So there you go. Now, cross sections were there one kind of difficult. Curvature was a whole other kind of difficult. So the oldest paper that I could find that talked about this was a paper from 1973. So basically what that paper did is it would flatten hair between 2 microscope slides and then use a transparency with arcs of different sizes to get curvature measurements. And so this paper in 1973 that I found, I was like, oh wow, this dude solved it, right? Like we have a thing that we can use. This is a funny story for another time, but I found out that my advisor was holding out on me and actually knew him. And I met him for the first time this year. He's very old, but he's great. And he was like, wow, I can't believe somebody cared enough about my undergraduate dissertation that I wrote in the 70s. I'm like, you've changed lives. I cite you all the time, you're great. Now, the issue basically that we have here is that hair curls in 3 dimensions, right? And so one of the things that you're doing to operationalize it with these methods is flattening it, which distorts it in a way. And comparing arcs, it was not fun. It's a lot of work. It's a lot of manual labor. So that all kind of goes back to, this is why people weren't doing it. It's not just people didn't understand that there's a better way. It's really hard. So I came up with a new lab-based protocol where I was able to overcome the 3-dimensional issue in another way, which is by cutting the hairs shorter. If you cut a curly hair short enough, it will only curl in 2 dimensions. So that helps not having to distort it, and you can operationalize curliness as the intrinsic curvature of the hair fiber. There's actually some interesting stuff there, if anybody needs projects, where there were some hairs that looked curly, but then when you cut them, the curl kind of disappeared, and that goes to show that there's like different ways of getting to the same trait. Another reason to phenotype carefully. So put them in tubes, you wash them, you put them in a petri dish, you take a picture. Great. And then image analysis part. So there were things you could do, software that you could use to measure the curvature, but it was a lot of manual work. So my undergrad was a lot of drawing circles on things on a computer. It is only slightly better than using transparencies, but it's just a lot of work. So you have these lab protocols, you've made that part easier, but if this is still gonna be a hindrance, people are not gonna wanna phenotype. So I was like, what if we automated it? And I wrote a Python image analysis program. Again, things that took very long, but I can summarize in one slide. I wrote one for both of them. So for the cross sections, the way it works is you just segment the image, binarize it, crop it, you have your little cropped image, and then you say, hey, Python, give me the properties. And for curvature, it was a little bit more difficult, but you end up getting the same binarized picture at some point once you've cleaned it enough. Then you turn it into this 1 pixel curve, which it can calculate all these properties for as well. So the thing that took forever was now automated. Great. Now you might be asking, it still feels a lot harder than just pointing at someone and being like, your hair's curly, your hair's wavy. Is it worth it? Now I hope I can convince you that that's the case. So that brings me to hair genetics. Let's start off with which populations have been of interest when it comes to the genetic study of hair morphology. From what I could find, most genetic associations were done in Europeans and East Asians with a few Southeast Asian samples. The studies that I found referring to the US or Australia would specify in one way or another that they were only considering people of European ancestry, so that's not much more. And then the most diverse sample that I could find was this 2016 study on the Candela cohort of various Latin American populations that were admixed. So that gave me some hope that the glaring omission of representation from the entire African continent could be rectified. It wasn't exactly what I was hoping for, unfortunately. And the reason is because of their phenotyping. When I looked at the methods, and I just hope if there's one thing you take away from this is that someone will read your methods. Write for that someone, please. Hair curliness was scored as straight, wavy, curly, or frizzy. And then I looked more closely. Few individuals were scored as having frizzy hair, 2.4%. And these individuals were excluded from the hair-shaped GWAS. Well, so the diversity that they had basically got thrown away because there's too few of them. And it made me think that they probably didn't have a lot of African ancestry in their sample either, which is kind of what you see here with their admixture plots. We can have a separate conversation about why that may or may not be, but at the end of the day, what this means is that there still isn't any great study of the genetics of hair in African and African descendant populations. But what is frizzy hair even? The best way to understand this descriptor is the way that it's most widely used in the GWAS literature, and it's as a racialized category referring to tightly curled hair considered to occur naturally only in African or other populations racialized as Black. So the idea that African frizzy hair is fundamentally different from other kinds of hair, including other kinds of curly hair, is reinforced by statements like this. Categories 1 and 2 define straight hair, type 3 defines wavy hair, types 4 to 5 refer to curly hair, typical for people of European ancestry. Okay, that seems like a weird extra to add to your phenotyping method. This is from a L'Oreal study, so shocker, people who are in cosmetics and develop products for hair think about this a lot, but they tried to develop a way of quantifying hair morphology by first measuring it and then creating a classification system. I would love to talk to people who measure things and maybe classify things or deal with people who classify things, because in some disciplines there's just this deep-seated desire to classify things even if you've measured them continuously first, and I don't know what that's about. But anyway, when you have things like that of you have curly hair typical for people of European ancestry, you've just undone all of the phenotyping. All of the value of the phenotyping is now gone because you've said, okay, well, only if you're this group can you have this phenotype? I would be happy to talk about why what they did was not great, but they basically didn't have a good justification for why each category is a category, right? Like you have a continuous trait, like where do you draw the beginning and the end of the different traits? And when you look at the types that are considered to be related to African and black hair, you see that actually it's pretty widely spread out if you're looking at a PCA of all of the variables that they measured. And so the question is always, well, why would we distinguish the difference between straight and wavy hair, but not distinguish the difference between things we don't have words for, hair? So, okay. Now, knowing that the reality of biology is beyond multidimensional continuous variation, one of the ways that we can step beyond the taxonomy problem is to look at people who don't fit in the taxonomies, looking at admixed populations. So I'm going to be telling you things that you already know, so let me know and I can skip through it. But if you're looking at a continuous trait where the mean phenotypic difference between two populations is non-overlapping, this can be like a great way to learn something about the genetics of the trait. This is something that's been done with skin color a lot, and so one of the things that you can do if you have a trait like this is to look at the correlation between the proportion of ancestry from one population and the trait that you're interested in, because the more, and I'm looking at Liliana because this is literally just what you were talking about yesterday, the more ancestry you have from population B, the closer you will be to population B in that phenotype. So this is what we see in skin color, and I was like, well, what if we did that in hair, because now we can measure it too. So with that in mind, I said I'd look at hair in African European admixed individuals. I started with a sample of 4,000 people. Then I had to find the ones who had at least 95% African and European ancestry. Then I needed to find the ones that had at least 10% African ancestry, because in our sample we had a lot of people with a little bit of African ancestry. And then ultimately, it came down to who did we have hair samples for? So my 4,000 sample came down to 193. And I had curvature for all of them, skin reflectance as a calibrating trait that we could look at alongside everything for a lot of them. And then for a good number of them, I was able to get cross sections as well. So first, I wanted to check that I didn't mess up everything. So are we going to see the same correlation that was demonstrated elsewhere between the proportion of African ancestry and skin color in my sample? Yes, awesome. We love significance. We love beautiful diagonal lines in science. So I knew that at least that was being replicated, and so I could ask the second question, which is what I was actually interested in. Do we see that same pattern with hair curvature? And yes, I was excited to find the same. It feels really silly that ultimately we just care about the shape of lines that we can draw, but it was a good starting point. It made me feel like there was a point to all of this effort in carefully measuring this phenotype. So to convince you that it was actually worthwhile, I want to show you what we were actually missing. So to compare with categorical typologies, this is what we were missing. In the same sample, and now I've just colored it by objective cutoffs for the different hair types, this is what our correlation was, and look at everything that is dark green. That's everything that was collapsed under frizzy or kinky. That's a lot of information that was being lost. And this is me being generous, because that is objective cutoffs. When you look at most GWAS, when you look at most databases, UK Biobank, looking at you, it's self-reported. So when we look at self-reported, you see how messy it really is. So on the x-axis, we have the objectively measured curvature of hair, going from straight to tightly curled. And then on the y-axis, we have different self-reported hair types from those individuals. We can all agree what straight hair is, and I think that that's beautiful. That's one point of agreement that we have. Wavy hair, a little bit different. Curly hair and very curly, I mean, what you consider curly or very curly, shocker, is really, really subjective. There's actually some interesting patterns in there relating to people's identities, how they racially identify, and also the percentage of African ancestry that they have. So if anyone's interested in anthropological analyses, that data set is waiting for someone. So another point I wanted to address was this idea of linking cross-section with curvature, right? So to recap, because African hair is stereotyped as having a flat cross-section, and Asian hair is stereotyped as having a round cross-section, people have extrapolated that, hey, maybe round equals straight and flat equals curly. So we are able to test this question in our admixed sample to some extent. If there's an intrinsic link between the cross-sectional shape of the hair and the curvature, we should be able to find it. So when we just look at it, we see, there's a correlation between eccentricity, so how round or flat the hair is, and curvature. But once you correct for ancestry, you see that that relationship is no longer significant. And that brings me back to this point of when we create qualitative categories and composite phenotypes, that is when we are moving towards racializing human variation. It may feel like a small nuance and who really cares when we're talking about hair, but let me illustrate it with skin color. If we look at my admixed sample and look at the relationship between melanin index, so how dark the skin is, and hair curvature, we see a relationship. So does that mean that the darker your skin is, the curlier your hair is, or vice versa? No, we know that that's not the case. But this can be really difficult to tease apart with traits that feel like they should be much closer to each other. And so going back to understanding the genetic basis of a trait, but also its underlying biology, it's really important to be able to tease apart traits that are going to be co-occurring in different populations. And chances are that there's just multiple traits that co-occur in different populations without necessarily being related to each other. So I'm just going to compare with previous GWAS now. There aren't many that were done including any African populations, but still you can learn something. And we were able to replicate 24 previously reported SNPs. So one of them, this is going to be an excellent example of cherry picking, was the SNP on chromosome 20, where previous studies reported that the C allele was associated with curlier hair. And in our sample, the C allele was associated with higher curvature. Wow, amazing. Now, what's great about this SNP is when we were looking at the allele frequencies, we saw exactly what we expected, right? The C allele associated with curlier hair is predominant across African populations, and then the T allele is fixed in some Eurasian populations, but like mostly present there. It's what you expect, because we were saying we're looking at this African European admixed population where the African population is associated with having curlier hair. But what was great is that we also found a high frequency of the C allele in Melanesians, which were not in our sample. And for those of you who don't know off the top of your head what Melanesians look like, they have very tightly curled hair. So these are the kinds of patterns that we'd be looking for to understand whether we're getting useful hits. So let's run into the current and future work. I spent all this time talking about measuring stuff and why that matters to our ability to understand the biology of the trait. But I'm going to talk to you about why that's going to keep being part of my work here. So I see my future and current work on human biological variation as integrating multiple scales of inquiry. So on the level of the individual, I'm really interested in the biophysical modeling of hair and skin. To do this, I'm exploring collaborations with people in computer graphics, which may sound strange, but when you think of all of the things that go into things like Unreal Engine, if that means anything to anyone here, and MetaHumans, there's a lot of people who are spending a lot of money to create realistic looking avatars. And as we're all preparing to be uploaded into the cloud, it's going to be important that we're well represented, right? So they have a lot of really interesting ways of measuring and simulating the optics of skin. But shockingly or not, they're really not good at not European skin. And part of the reason is the way that they think about getting data from the full range of human variation. So these are spectral reflectance data from different populations from a computer graphics textbook. And basically they have African Americans, East Indians, Asians, and Caucasians. If we got all of these, we simulate all the people. I don't even know what their sample size is. I'm pretty sure that they might have had like 1 or 2 people from each, because the funny thing about computer graphics labs is they tend to do all these studies on the people in their lab. It'll be like, wow, no I or B, just nothing, just come here. But anyway, in anthropology and people who've done skin color genetics, like we have lots of reflectance data. So one of the things I'm working on right now is trying to use full spectrum reflectance data to understand how that's related to skin color, and also if we can develop baselines for people outside of genetics and anthropology to make sure they're inclusive of the full range of human variation. Because again, if you're relying on this unclear description of what different groups of humans are, you're probably missing a lot of variation. As a side note, facial recognition, a very big interest of mine now, because that's another group of people outside of anthropology, genetics, biology, that are working with something that so desperately requires an understanding of biological variation, but are not engaging with any of us to, big detriment. So there's a lot of issues of the performance of facial recognition algorithms between different groups, and again, it's like, how are you defining those groups? Ultimately, the issue is they have bad training data sets, and they have bad ways of defining how their algorithm is performing. That's the concise version. Okay, and then one of the other things that I'm doing is trying to go beyond comparing or relating the phenotypic to the genomic by thinking about all the intermediate steps. How can you break down the phenotype? Further and further, how can you get to the point where you're thinking about the biological pathways and the complexities of everything that is happening on a genomic level, which is where we get to hierarchies and systems. Now, it might sound overwhelming to do all of this, but some people have done it. This is a really cool paper that I found that was a machine learning model that described a phenotype-genotype association that hadn't been shown empirically yet, and it was later confirmed. So they were able to predict something new. Yeah, it was in yeast, but it's still cool. And there's other people who are doing work in this realm relating to humans, and this has to do with how we can use information that we already have on gene sets and pathways to not be just doing a bunch of linear regressions across the genome. So I'm working with a master's student at North Carolina A&T to do this, and Lauren Crawford at Brown. And basically, the last point of this that I wanted to make is all of this information allows me to break down complex phenotypes into components that can be studied in various combinations on a population level. Advanced graphics, very impressive, right? So the final branch of research I want to talk about is putting all of this together towards my ultimate goal, which is understanding everything in an evolutionary context. And this is where I also am doing some work on sex as a dimension of variation. This right here is a type of langur. What kind of langur? Dusky langur. That has a baby here that clearly is a very different color, and that's like natal coats. So what's really cool about hair and sex is that hair allows you to think about sex as this dynamic change across your life, because depending on hormones and a bunch of other factors, the same genome will be used to express different things. And this is work that my student Sarah is doing, and we're looking across the entire order of primates at how natal coats are co-occurring with sexual dichromatism and other things. So, to conclude, so far, even though my work has focused on hair and skin, I see those as entry points into a broader discussion about racialized human variation. I'd say that historically, this is what has been done, but clearly we still do it today. I maintain that Linnaeus is to blame because, maybe controversial, I don't think it is, taxonomy is fundamentally incompatible with the dynamic nature of evolution. So if we're constantly trying to put things into little buckets and classifications, we're just constantly going to be running into the same issue. And there is no evolution without variation, but there is no taxonomy without classification. So I really think fundamentally we need to step beyond classification. So the takeaway is race doesn't just persist because of ignorance. The call is coming from inside the house. We need to fix the way that we are talking about trait variation and measuring it. Hair and skin are useful traits to explore racialization versus biology, also sex. And if you're interested in a collab, let me know. So just want to thank all my collaborators and take any questions you may have.
Okay, yeah, we have time for a bunch of questions. So take it away. All right, I'll get it started selfishly. So I was curious with historical methods for phenotyping different cross-section things, is it harder to do a good job if hair has a higher curvature?
Yep.
Because like you're more likely to get
Yep.
an angle that's not perpendicular to
Yeah.
the tangent?
Yep, that's one of the main issues. And so when you go back, it's just a lot of people complaining about that. But the consequence is also a lot of people then avoiding doing that work on curly hair because they're like, whatever. And then you have this question of like, how do you know how messed up your angle is? You don't.
Right.
So yeah, just not great. And there's a lot of weird methods as well. There's also people who use shrink wrap. Like you thread a hair through shrink wrap and then there's a lot of stuff out there. People have tried.
How do you control for like full head variation in hair curvature and stuff?
That is a great question.
My hairs are very curly.
Exactly. That is a fantastic question. This is exciting to me because I'm like, these are the questions that we should be asking. Like whenever I see a GWAS, I'm like, how did you operationalize it? What about within individual variation? Within individual variation should be included in the analysis. So ultimately the idea would be to get, as much hair data about an individual as possible, but also that's when we get to some of the cool work that I want to start doing on gene expression. So there's a collaborator in MCDB here who I'm going to bully into doing some gene expression stuff for me, where you can imagine looking at someone's eyebrows and their scalp hair, right? Like why? Just why? Question mark. That's ways of like answering that question. Like what is being expressed differently in order to make the phenotype different?
Just a follow-up, I'm just imagining that if you took all the hair, which would be millions probably from a head, has anybody looked at how much variation there is within an entire head of hair?
That is a great question, and yes. So during my PhD, I overlapped with a previous student who used to be an FBI fiber analyst. And so before she joined our lab, one of the things that she was doing was racial origin determination and all that kind of stuff. And once she was in our lab, she did a lot of measurements and she just did cross sections. And there's more variation within an individual scalp than between individuals in a lot of cases, which is one of the many reasons why it is not advisable to try and individuate someone on the basis of hair, and why the FBI has overturned like 98% of the cases that they've done with like hair evidence. So yeah, not great.
I was wondering, because you mentioned variation and sex at the end, if that was a factor in the samples you were using, like were you using samples for hair from like a variety of sexes, or does that matter? Like, is there evidence to show that it doesn't matter? How did that factor into the methods?
That is a great question. So actually there's two answers to this. So I started doing this in 2011. And so the first paper that I published on this was in 2016, and it was hairs that I collected from people that I talked into giving me hair samples while I was an undergrad. So I knew every single person. I can recognize them from their hair. And it was a bunch of ANOVAs. I didn't have genetic data, right? And that's a really big interest of mine. How do the methods that we use to analyze data limit the questions that we can ask? And back then it was like, well, if you're gonna do that, you need to make sure that you're analyzing males and females separately and all of that. But now we have much more sophisticated statistical methods that we could theoretically take all of that into consideration. So one answer to your question is we included sex as a variable in the GWAS. Another answer to your question is we want to move beyond needing to define sex in that binary way. Haven't figured out exactly how, but part of that is going to have to do with within individual hair variation, because there's going to be differences in how where you have hormone receptors in your body, right? And so that can already be the question of sex, again, in this dynamic way that's not just a binary. Yep.
I'm trying to ask this question without using the phrase harebrained or cutting edge. But what is the craziest sort of out there new method that you or others are thinking about how to go from your friend's hair to taking these measurements? Like, can you 3D print something? Can you use a laser on someone's head? I have no idea. What have you thought about?
No, those are great questions. So actually, one of the things I'm trying to do while I'm waiting for my lab to get built is travel to some previous collaborators who have access to a micro CT. So I came from bone people, which sounds weird to say, but all of the measurements of bones that they do, that inspired me wanting to do all of those things. And so the idea would be to get like 3D volume of individual hair fibers, but then you kind of get to this issue of how do you analyze that data? And that's, again, this issue of like, are our statistical methods and the way that we're thinking about analyzing the data limiting what we can do? Can we just throw it into the AI machine and it will tell us? No, but there are more sophisticated things that we can do. The issue is then also trying to take away the effect of whatever someone is doing with their hair, even the length of their hair, when they last washed it and all of that. Although we can theoretically get that information. So my goal would be to, at the very least, develop a computer vision program where you can just take a picture of people's hair and infer what the individual intrinsic curvature of their hair fibers would be, but that would involve us having to do that with samples where we had the hair samples and had also just like pictures of people and see if it's possible to infer that relationship. I think that's the craziest. A lot of the people who are in like dermatology and cosmetics, I don't know, they just don't do really crazy things. I think it's because they have stuff to do and like money to make and patients to treat. So they are, and I kind of joke, but even skin reflectance, I presented that as clearly we have the answer. In dermatology, they use this thing called the Fitzpatrick scale, which is garbage, and it's subjectively defined. And I'm always like, but you could just, and for some reason, I don't know, like that's one step too much. And so I think there's also a lot to learn, and that's more of the anthropological STS questions I want to ask. Why do people not do the right thing? Why do people use the methods that they use?
How much does natural selection explain the variation in these characteristics of hair that you observe, and how much might be genetic drift?
That is a great question. So what inspired me to do all of this stuff was seeing the classical maps of the distribution of skin color and the distribution of UV radiation. I was like, whoa, mind blown. What about hair curl? Like, is it the same thing? And the quick answer is no. The distribution doesn't follow any one climatic variable, but the working hypothesis for like me and my advisor is that early on in human evolution, so early Homo erectus, let's just say like 2 million to like 1.5 million years ago, you have fully bipedal hominin. We have reasons to believe why that might have been the point at which we lost body hair, we're sweating, we're running. That might have been the point at which it became beneficial to have scalp hair, and specifically tightly curled scalp hair. And so the one thing I didn't talk about is the work that I did showing heat transfer through different hair textures. And basically, tightly curled hair is able to create maximum distance between the scalp and the top of the hair in a way that allows you to lose maximum heat while minimizing, specifically from solar radiation, how much heat gets to your scalp. And I emphasize the solar radiation part because if ambient temperatures are high and it's humid, you're not gonna sweat. The Amazon, there's nothing you just gotta be naked in the Amazon. But if you're in the desert and it's super sunny, then having some kind of block would help. And one thing that we see is in the sequence of traits of human evolution, brain size comes last, which was historically kind of a surprise for people. And so there's this question that's still outstanding of what was the reason why we had, fully bipedal look like us type humans, but then brain size was still not where it is today until later on in Homo erectus. And maybe that was the reason. And so then it's possible that it was under selection early on in human evolution, and then the rest is drift, is the short version of what we think happened.
Tim, I think that there's a lot of natural selection for no hair. We're at the forefront.
Sorry, I have one more. I recently was told by someone, and they could be wrong, this is just like a scientist only thing, but that white skin evolved the most recently. And is that true? And what can you tell me about that?
I have so many questions. I have been on the terrible side of the internet for a very long time because of the things that I study. And so that already given me hints of, ooh. I remember seeing a lot of things where white supremacists were arguing that Europeans were more evolved. It's kind of where so it's everyone is equally evolved, we're all modern humans, right? But one way of answering that question is this the most derived trait? Is light skin the newest iPhone model, whatever? No. There are different populations that have gone through selection for dark skin again. So we see that, and that's why we have this really great pattern of following UV radiation, because populations were being exposed to different UV regimes. And so there are going to be some populations who recently evolved darker skin, is the long and short of it.
Okay, well let's all, one more question.
How does the hair change over the lifetime in development? Have you been thinking with hormones?
That is such a good question. And that's why I'm super excited about all the stuff that Sarah is doing. It changes a lot in a lot of different ways. One of the fun things about doing a lot of public outreach is that when you have a contact form, people will send you the funniest stuff. So far, nothing awful. I'll just put it out there. But people will be oh, this is how my hair changed. And a lot of people tell me I was born with dark hair and then it became blonde. I was born with blonde hair, it became dark. I was born with straight hair, it became curly, all of these things. And especially a lot of women, specifically, I remember saying that their hair changed after chemo and after pregnancy. So we know that, right? Send me people who can analyze things and we will have answers to your questions. So that's kind of where we are. It's there's all of these questions to answer, so I'm really serious. If somebody's oh, I really care about hair and skin, wants to do research, I'm here. I'm just 5 minutes away. Come see me.
Yeah, so I'll give Tina a big hand.